Good morning, everyone, and good afternoon to our listeners from Europe. This is Aisha Sinchodhary from Cytel. On behalf of Cytel, it's my pleasure to welcome you to this morning's webinar on synthetic controls in clinical trials. It's my pleasure to introduce Christian Thorland. Christian, as many of you know, is one of the top 1% of highly cited researchers internationally in all fields related to biostatistics. He is a professor of biostatistics at McMaster University and a senior vice president of real-world evidence at Cytel. So with no further ado, Christian, let me turn it over to you. This is a talk that's going to be based on both the ebook that most of you received when you registered, as well as a new primer that Christian and his team have developed. So thank you very much, Christian.
Okay, so I figured before we start talking about examples of synthetic control arms, applications successful or not successful, it'd probably be good to just go through where it's actually appropriate to use them. So here I've listed a few. There are probably a couple of others that I'm missing, special cases. But I think the the 4 cases where we're seeing it the most are rare diseases, as most of you, if not all of you, probably know, where getting a control group is simply quite difficult often, or even randomizing to control is next to impossible, simply because the disease is so rare, so it's challenging to get patients. I think they're starting to be used a fair bit as kind of more of an internal tool to see if you can get around randomizing fewer patients to control, simply for issues of difficulty in recruitment. You've certainly seen that with the latest COVID pandemic, where there's just an excessive demand for patients. I think there are over 1,000 clinical trials planned right now, and with their planned sample sizes, they're likely to go up to, well, I think close to almost all of the cases that have been documented thus far, which is a bit unrealistic. So one really has to wonder, do you really need to enroll all those patients into a control group, or could you just take that data from somewhere else? And of course, there's similar issues in many other disease areas, rheumatoid arthritis, psoriatic arthritis, other autoimmune rheumatic diseases are often having difficulty enrolling patients. Cancer trials as well, and it's partly because patients are getting better, so it's tough to get representative populations, and partly because there's a fear of getting into a controlled treatment and not getting the optimal intervention. And a bit to that point, I think a lot of clinical trials are also starting to incorporate, sometimes planned and sometimes it's involuntary, that you kind of have to switch placebo to the more active intervention into the trial, whether it be after maybe 3 or 6 months. If the patient's failing on placebo standard of care, you simply have to switch that patient to the active intervention, and for that reason you don't get long-term data. Or sometimes sometimes it's planned and sometimes it just happens. And of course, we have we all know the notion of clinical equipoise, the state that we have good enough or large enough uncertainty about the efficacy of the experimental intervention that we're okay to randomly assign patients one-to-one. But equipoise is really a term that it was very valid, I'd say, 20 years ago, but the way that medical research and medicine's kind of shaped over the last couple of years, it's questionable in a lot of situations where you do clinical trials. So those are some of the main reasons and situations where it's appropriate to do synthetic controls. And this talk won't be a full hour, so there's certainly time to discuss other situations at the end of it. So before I dive into it, I'll say we did publish a primer on this fairly recently. And so she mentioned that, and the ebook that's been shared with you, it's a bit more of a lighter version of this. This is a bit of a heavy academic reading. So I think we did intend this to be going through the primer step by step, but to be honest, it's a lot of text and it's a little boring and it's kind of a checklist. And that's not to say you shouldn't read it, but you know, it's if you want to get into the details, and I kind of feel like for a talk, like it'd be a bit more interesting to go through what examples actually work and what examples don't. So without further ado here, I'll go into our non-bullet points outline for this talk. And so what I want to start with is a couple of what I'd call success stories, you know, as good as can be stories, because synthetic controls are really never perfect. I think it's kind of an illusion when people talk about, oh, you can just use real world evidence and then you'll come up with a control group that's as good as if you didn't randomize. That's just not true. But there are a lot of stories out there, a lot of cases where the use of synthetic controls were actually quite helpful. It did actually solve a problem that couldn't otherwise be solved in a fairly short period of time or with the available resources. So I'll go through two of those. First, one is with respect to regulatory purposes, so the story of blinatumomab. Secondly, I'll go through an example we did ourselves. It's also in a publication. It was more of an academic exercise on how you can actually use synthetic controls to save resources, essentially. So money, time, and just generally gain efficiency. And then I'll go down to what I think is probably more common, some examples of where the data and the setup is a bit more challenging. It's really very far from perfect, but it's also not so bad that you can't do anything. I'll go through two examples of that, and at the end I'll talk a little bit about you know, subgroup effects as one example of how synthetic controls often hold a lot of promise, but you have to exercise a lot of caution and maybe just cut your losses in due time rather than keep pursuing it. Okay, so I'll get started with the success story of blinatumomab. Let me just give me 1 second here. Okay, so blinatumomab was it's a BiTE antibody, it's bidirectional antibody, and it received accelerated approval by the FDA in 2014 based on use of synthetic controls. And the data that they had for blinatumomab at the time from clinical trials was simply a single-arm phase 2 trial with 189 patients. But what they managed to do was to get an accelerated approval based on use of synthetic control arms from clinical trial data on standard of care from actually 11 databases, US and European, for complete response and overall survival. Again, data from other clinical trials. So it really is data that's collected in a very similar setting to the setting that the phase 2 clinical trial was. And it's collected with the purpose of research, and patients are randomized. Of course, you lose randomization when you're just taking a control arm out of a randomized trial. But it's kind of as good as it can get with respect to synthetic control arms. And so then they used this data and estimated the control rate and the comparative treatment effects on complete response and overall survival and a number of different outcomes. And the way they did this was quite exhaustive. So a lot of different analyses that went into it. This slide and the next slide is just a quick excerpt of some of what they did. To begin with, they did a number of stratified and weighted analyses, split up by disease status and age, kind of looked at how comparable the control rates, this is just for a complete response, looked at how comparable the control rates were across those subgroups and across the two intervention groups as well. And then did a weighted analysis. They did the same thing for survival outcomes, and they did a whole bunch of statistical analyses with a lot of sensitivity analyses to it, inverse propensity for treatment weighting with and without adjustments, and I forget what the S stands for here, but something else. And what they found was not surprising, because there's quite a lot of data that went into this. So when you have a lot of data, you typically don't get a whole lot of sensitivity on the various statistical models and analysis methods that you use. So what they were able to show was essentially that there was an increased odds of complete response of blinatumomab, and a lowered risk of death, also with blinatumomab, and all the different sensitivity analyses were quite consistent. And what's then worth noting is that the approval was contingent on the success of a later phase 3 study, which came out in 2017 and also showed a statistically significant benefit. Here's just the overall survival results. And what you'll see, I'll just go back here. What you'll see is that the hazard ratios are around 0.5, so it's a 50%-ish reduction in death, and that's a pretty large effect. I think with all things observational, synthetic controls, you have to treat them as an observational study. There can be a lot of biases, but even if in an observational study, if you have a very large effect, you have to recognize that there might be some degree of bias, but if the effect is very large, probably some of that is due to a treatment effect as well. And so I think what you're seeing here is probably that the phase 3 clinical trial was giving you a result that was much closer to the truth. And so you're getting a 30% reduction in death, as opposed to a 50% reduction in death. But again, it shows a signal very much in the same direction. It is quite compellingly statistically significant. So based on that, there's more to that story. I'm really just putting out the simple stats here. But based on that, the approval was then continued and not discontinued, which is a good thing. I think one of the key things to hone in on here is that some of the reasons that they were successful with the use of synthetic controls here was manifold. First of all, there's an appropriateness of the indication, and they had a proper identification. There's a lot more to this background story. They had a proper identification of the external controls that they used. They all came from clinical trials that were sufficiently similar. And lastly, they used a lot of different statistical analyses, a lot of sensitivity analysis. And the other thing that's also quite key here is that you need to test the robustness. And so one of the things is whether it's propensity weighting or Bayesian commensurate priors or other methods that you want to use, it is necessary to test the sensitivity or the robustness of that, because if you start getting different results from different models, it's a little tough to trust, particularly for the FDA and EMA and other regulatory agencies. Okay, so I'll go on to the next example, and that example is not something that was used for regulatory purposes, but it was really something that we just did for internal digging, and we published a paper on this, and we've done a bit more work, and so presenting a little bit of a mix of results there. And that's using what is called dynamic borrowing in the Bayesian framework, which is essentially that you take external control data. It can come from multiple sources, a bit like you saw with the blinatumomab example, and you create a model where the external control data is used as kind of a prior distribution. You mix that with the likelihood. This is a very oversimplified way of what really happens in the background. There's a lot of Greek letters and formulas and what have you that I just don't like those on slides, so I won't go into statistical details on that. But you essentially take the external data, turn it into some sort of prior belief, and mix that with the data, which is the likelihood. And then, as with all things in the Bayesian framework, you get a posterior distribution that you can draw inferences from. And what this really, where this really benefits clinical trials, I think, is that instead of randomizing 1-to-1, you can actually reduce your randomization to control. And so for the reasons that we talked about earlier, which is that it is either it can either be too challenging to enroll patients, so every patient counts, and the more patients you can get on your treatment, the better. Second of all, there are also issues sometimes with patients don't wanting to get onto clinical trials simply because they want to get the experimental treatment, and the outlook of having a 50-50 chance of just being given the standard of care versus a new experimental treatment when you're kind of at your last hope. It's particularly the case with oncology patients, a lot of dire oncology prognoses. So what you can really do here is that you can get a randomized clinical trial that is randomized, because it is randomizing patients to control, but it's not randomizing at the same ratio. It's randomizing a lot fewer patients, so that there will be a lot more willingness for patients to engage and enroll in this clinical trial. But at the same time, you can combine the external data with the concurrent controls that you're randomizing to. And ideally, there's some balance in here with how much data you need and what's too little. Ideally, you should be able to get the same statistical precision and accuracy and reliability of the control group, and basically get the statistical inferences that would have the same strength as if you had just done a one-to-one randomized trial with the same amount of patients that ended up in the treatment group. So what we did is we went played around a bit with numbers. There's an amazing initiative called Project Datasphere, and if you ever want to look at individual patient level data from clinical trials and play around with this, I'd highly encourage you to go look at it. You can go and get data from it. It takes a little bit. It's a little bit of an approval process, but I think compared to most other data sharing initiatives, this is by far the easiest to use and most useful I've ever come across. There might be others that I don't know of. And so what we did here is that we took a number of clinical trials, 4 clinical trials to be more specific, that looked at treatments for stage 3 and 4 non-small cell lung cancer. And we did a lot of different analyses, but the one I'll be talking about here is taking the INTEREST trial, which is the second column, as the anchor clinical trial, and then trying to pretend as if we were running that trial with a different randomization ratio, so enrolling fewer patients to control, more to treatment, and then using the control data from the other 3 trials to see if we can get the same results, and how much control data was needed to get the same precision, as well as when do we run into trouble in terms of the control data not being sufficiently similar. And you'll find you can see the PROCLAIM study is quite a bit different in that it has 100% stage 3 cancer patients and 0% stage 4, whereas the other trials have, well, varying degree, but more than 50% of stage 4. And there's a number of other differences. For example, the median overall survival is much in the control group is much larger in PROCLAIM than it is in the other trials. So the PROCLAIM data was kind of our a bad trial, and we kind of wanted to see what happened if we mixed that into it. And so what we did here is that we took the INTEREST trial, used that as an anchor, played around with a number of different scenarios. This is by no means a talk on this particular project, so I'm going into much more detail on it. But we played around with a number of different randomization ratios, so 1:1, 1:2, 1:4. I think we did 1:5 as well, and 1:10. I'm just showing you these for simplicity. But what we found is that at least for this particular case, going down to a randomization ratio of 1:4 gave us both a control group estimate as well as a hazard ratio estimate that was quite congruent, that yielded quite congruent statistical inferences with the results of the original trial. And so we tested this for a number of different scenarios, and like I said, kind of using PROCLAIM or some of that data, throw them in the mix to see what happens when your data is a bit more heterogeneous. We even flipped this around and let the PROCLAIM data be the anchor trial to kind of saw what happened there. But the conclusion of that was that in most cases, if you're using these Bayesian dynamic boring methods, which again I'm not getting into detail of the statistical details of it, but if you're using them, and probably the same is true for propensity weighting and other boring methods, then you should be able to reduce your sample size in the control arm by up to around 60% without any meaningful loss of validity of the statistical inferences, as long as your data is only heterogeneous but not totally inconsistent. And so it's a little, how do you judge heterogeneous versus inconsistent? Well, just that there are some differences, but they're not completely different direction of effect, and it's not like one control group is 5% and the other one's 25%. It's a little subjective, but in our case, it's at least we show that it works even if you have a bit of heterogeneity in your trial. Okay, so the good news about this is also that the FDA is recognizing dynamic boring, particularly Bayesian methods, and what you will hear called in more statsy talks, commensurate priors, and there's a number of different methods that go under that label. And that's all now approved and endorsed by FDA and I think also EMA. So all this is kind of moving in the right direction, so to speak. Okay, so I'll go on to the next. Now we're getting to the moderately challenging data scenarios, as I call it. And I'll go through a couple of. I won't really get into the details of exact examples. We've done a bit of work, but there's still have confidentiality there. So I'm just going to talk conceptual about some of the things we've seen in the past. I think the first thing to note is the treatment switching, which is something that happens quite often in various autoimmune diseases, particularly when you have biologics on the table and patients have kind of, multiple lines of treatment for chronic disease, autoimmune diseases, and patients have kind of failed everything that's available up to the point of getting a biologic. You can't really keep them off a biologic for too long, for again, ethical reasons, and also because then the patient just doesn't want to be in the randomized clinical trial. So what often happens with these trials is that there is an option for placebo, folks in the placebo group or standard of care group, to switch on to the active intervention group. And it can be one of many active interventions, or it could just be a 2-arm trial. And what that results in is you either end up with a very low number of true placebo patients, or you end up with a reduced duration of placebo. For example, if they're all switched after 3 to 6 months, but you're really looking at 1-year results. And the same, like I mentioned, the same thing is true in cancers with poor prognosis. So, for example, probability of 1-year survival is very low. You definitely want to get them onto a treatment that works as soon as you can. So that this type of scenario created a few challenges, and I think I haven't seen it used so much personally in regulatory instances. If someone in the audience has, I think it'd be great to if you can share that example in the chat. But where I've seen it applied a couple of times, we played around with a few things these ourselves. It's never really went anywhere for internal corporate reasons for clients that we work with. But essentially, it's something like if you're dealing with a survival curve in cancer and the control group just isn't there because they had to switch it, or you just can't rely on it. Patients went somewhere else that dropped out because they're feeling sick, and you have some historical control group data from previous trials that are probably a couple of years old because it's before this experimental treatment came out. And just trying to use those together, I think is the best option. It's essentially like you don't have a control group, although you do have a bit of, you have to start so you can stabilize the estimation, but it's really not perfect, which synthetic controls never are. Okay, I think the next example, I won't go that much into specific examples. I will mention one, but one review that I'd like to highlight here was this paper by Baumfeld and Andre, which I just think is a brilliant review of how real-world data has been used in clinical trial designs in the regulatory landscape. And they've looked at 22 submissions to the EMA and FDA, and what you'll see if you give a read or just look at table 2 in the paper is that the vast majority of these are actually synthetic controls, and the vast majority of these are also rare diseases. And so it's essentially just not just because it's a bit more complex than that, but it's essentially examples of single-arm clinical trials, typically less than 100 patients, and then some use of historical controls or natural history studies to kind of pair those up, because that's simply the only option. I think one example from there that I would like to highlight is the example of cerulopinase alpha. It's used for the treatment of Batten disease, and it's a quite brutal degenerative nervous system disorder that happens in quite young children. So the fact, and I think that from what I recall, the story here is that there basically hadn't been any good new treatments for the last 30 years, and then Biomarin came along with their Brineura, and it really made a difference for a lot of kids. And it's a pretty fascinating story. There's even a whole documentary on it, so that just kind of speaks to the gravity of this. The data scenario was that they had a single arm trial with 22 patients. It is a rare disease after all. And they had a well-defined historical natural history cohort of 42 patients. So it's not exactly the sample sizes that you can use to do run advanced or sophisticated statistical adjustments. It really just becomes a what's the mean in one group versus what's the other. But I think it speaks very much to the point that, because I think a lot of time when we talk about synthetic controls, we'll talk about propensity scoring or even AI or real world evidence, and there's a lot of promise of all these fancy methods that we can use. But the truth is that if the story is compelling enough, and in this case, I mean, if you don't know about this example, I'd strongly encourage you to go look into it, because it is quite amazing what this treatment does, and sort of how life-changing it's been. I'm not advertising for Biomarin here, but you don't need to have sophisticated analyses, and you don't need to have super compelling hundreds of patients of historical control data if the story is compelling enough. And probably what really went into what went into this is that the discussions that went on before and just the importance of the topic is what got this approved, contingent obviously of the availability of some basic data. So again, complexity is not always equal to success. Sometimes it's just take what you have, make sure that your story is compelling enough. Okay, so I'll get on to the last, and so we kind of start at the top and end at the bottom of where you probably should not be doing synthetic controls. There is a plethora of examples where I think you shouldn't. Let me just give you one, which is, again, it's kind of an amalgamation of various similar topics that we've seen before working with both pharmaceutical companies and also academic endeavors, where the goal is really to compare one new treatment or just one sort of off-label treatment or what have you in one population, but most of the data is on another population. So what you can imagine is, let's say one pharmaceutical company has a fairly small randomized clinical trial that they decided to do in an Asian population on a semi-rare subgroup. Could be some genetic marker or maybe some line of treatment, late stage poor prognosis of cancer. And they really want to, and some competitors beat them to it in the non-Asian market and also already approved in the Asian market. But this company thought, oh, let's do it in the Asian market because at least we can get in there. And then now we've run out of time or resources, so let's try to see if we can use this data to also get indication for the non-Asian market. And the evidence is just completely split. You can't do an indirect comparison of the two randomized trials because they're completely different populations. You have a little bit of single arm evidence in the overall population for the non-subgroup Asian population. You have a little bit of single arm evidence in the non-Asian overall population. But it's a bit of a mix, and reporting really isn't good. You can't really get access to IPD with the exception of your own clinical trial. The trial is so small that you can't really do proper propensity scoring. So you can kind of see where I'm going with this. And I think what is also important with synthetic controls is to really realize that oftentimes it's just not worth it. Like if you look at the data, if you do initial scoping review, you do literature review, subgroup reporting is not great, availability of individual patient level data is not great. You kind of play around all options. If nothing seems to work, then just cut your efforts and try something else. I know that's challenging if you're a biotech and you have one product and you have to do something, but if you're a larger pharmaceutical company, if you're working with a larger pharmaceutical company, there's an opportunity cost to pursuing this. I think in every instance you have to kind of consider is if this is not yielding any results after 2 to 3 months of comprehensive literature reviews and exploring different propensity weighting, and they all kind of give the same not so promising results, then it's probably better to just cut it than to continue with this for an extra year or two, which unfortunately I've seen a few too many examples of. So I think I won't give you any specifics on this, but I think this is a pretty common problem to see, in particular all the subspace indications that are being applied for in oncology. Okay, so getting back to the primer, this is a simplified version of one of the figures we have in that, and it's kind of just summarizing what we've learned and what we discuss in the primer. And this is a conclusion slide, by the way. So I don't like bullet points that much, as you can probably tell. But just to summarize in general, what makes a good synthetic control, or an as good as it can get synthetic control, and what makes a not so good synthetic control. And it's not rocket science. Your data sources have to be well defined. Ideally, if they come from other randomized clinical trials, which I think there's so much data from other randomized clinical trials, particularly within large pharma, where you have these large programs of oncology and there's systems being set up to access internal IPD. There's almost always not almost always, but very often there's a very good source of internal data, rather than the data you can get from, let's say, real world data vendors that are maybe a bit messy and not designed for research purposes. Of course, there has to be an effort into verifying the similarity of the data sets with the experimental single treatment arm, or the randomized trial with lower randomization to control, where you want to augment the control group. And there's a number of examples that we've gone through of where things are maybe not so similar. Of course, the methods of analyses have to be appropriate, but again, sometimes it's just the case. Sometimes you don't really need complex methods. But if you're not in the situation of a rare disease that is, especially pediatric rare disease, that really is prioritized by regulatory authorities, and you just need to show a bit of decent data to get the approval. If you're not in that situation, I'd say always try to aim for as high sample sizes as you can, because that's really where you get the opportunity to make some adjustments. They may not be the fanciest in the world, but at least you can test a lot of sensitivities, and you can test the robustness of like the sources of control data if you're looking at multiple sources, and you can really look into where is it that you're, what part of the external control data that you're considering is it that you can use for this particular exercise. And I think in the synthetic control projects that I've been part of, we always start with a big scoping review of what's all the data that you can work with. It's not like you just have a set of data to begin with, just use that, okay, here's your control data. There's always a bit of a process that goes into it, where you do a lit review, you look into what are the sources of where you might be able to get individual patient level data, might get some good covariate data. And so the more you can kind of up the sample size, you can at least start exploring, and then you might cut something out eventually. But if part of your validity or reliability argument lies on the argument of statistical adjustments, then you have to aim for sample size, because if you only have 100 patients, you can barely even reliably adjust for one variable. And then, of course, the relevance, and you have to make sure that there are no apparent biases in the trials or the data sources that you're taking. So oftentimes you will have a number of recorded variables, but are there other variables that are not recorded that might show the unknown biases? You have to make sure you have an adequate number of covariates. Typically, when we aim for high sample sizes, then we later on learn that the number of covariates that are available are maybe not that high, or the way that covariates are recorded are not the same way as they are in our experimental treatment data, and so forth. So it can be all kinds of challenges that make these synthetic controls not feasible. Again, I would say this, of course, it's complex. It's a complex process, but it really is. It's not rocket science, right? It's more like meta-analysis of observational data. And for anyone who has studied epidemiology, which I assume pretty much everyone on this call to some extent, will know what makes a good study and what doesn't make a good study. So I think we should there's a lot of promise around synthetic controls. There's a lot of times where it can go wrong. I've done a fair amount of these by now, and I'd say my lesson that I've taken away from this is continue where it makes sense, but there are a lot of times where it doesn't always make sense. Just cut your losses and think about better clinical trial designs there. So I'll end it at that, and I hope you guys have some great questions. But otherwise, thank you for your attention.
So Christian, the first set of questions that seems to have come in is about calculating sample sizes using SCA methods. And the first question is very general. Could you say something about the sample of patients that's needed and what kinds of methods correspond with which sample sizes?
That is that's a very good question. I almost think it's well, it depends on what situation you're in, because one where it's probably not that relevant, or it's not the most relevant thing, is you know, in rare diseases, because you can only get the data that you can get. So there's no point in doing a sample size calculation there. I think where it's probably the most relevant, and I would almost say it's not actually an issue of sample sizes, it's more an issue of simulations in a similar way that you would do with adaptive clinical trials. Because you know, I think sample size calculations are a little bit of a hoax, right? You put in all these assumptions like, oh, my control group is going to be this, my intervention effect is going to be this and this, and my dropout is going to be that. And it's just, most of it's pure guesswork. Not the control group risk, maybe if you have a comparable control group, and that, you know, that's kind of the argument for also doing a synthetic control. But your whole, you know, your treatment effect and dropout, and you know, if you knew that, you wouldn't be doing a clinical trial. And I guess I get that the argument is, well, we're just looking for a minimal important effect, and that's what we're aiming for. But typically, that requires very large sample sizes. So I think when you are in the situation of having to use synthetic controls, it's like either you're in that situation where it's just, you just can't enroll controls, and that's typically in the rare disease space, or it's just not ethical. So that in that case, you wouldn't need to talk about sample sizes. I think if you're looking at having uneven randomization, so like 1-to-4 randomization ratio, and then using external controls with, let's say, commensurate priors. I don't know of any sample size calculations that inform that, but I think what you rather would need to do is to basically simulate a number of different clinical trial data sets, or if you have some data sets that you're looking at already, then simulate what your clinical trial might look like under that randomization ratio. You can test 1-to-2, 1-to-3, 1-to-4, 1-to-5 randomization ratio, and under certain assumptions of the treatment effect, or you know, certain biases with covariates and what have you, you can simulate what's actually going to happen when you use different methods of, whether it's commensurate priors or propensity scoring or what have you. I think clinical trials in general, not necessarily rare disease single arm trials, but randomized clinical trials in general are going more towards simulating outcomes under different scenarios rather than the fixed sample size calculation. And you know, you've seen this a lot with COVID, whereas a lot, you know, talk about adaptive trials and what have you in the media, and no one's really thinking fixed sample size anymore. Well, there might be a few, but I don't think it's the right thing to do in a situation where things are moving so dynamically. So I would say it's a simulation problem. If you're actually randomizing some patients, even if you're not, then it's still a simulation problem. And sample sizes of sample size calculations for regression models is there's a lot of papers on it, but again, I think simulation is going to give you the best answer, because otherwise it's pure guesswork. You have to test a lot of different scenarios.
Right, great. So this actually leads a little bit to the next question, which just asks about how do you calculate sample size? What are the methods when you have a situation where the control is constantly changing? How do you go about that?
Again, my answer would be the same. Actually, I think it's I would simulate it. I don't think it's not my area of expertise, sample size, but changing control groups, and I don't think it's a good topic in general. That's my opinion. I think again simulations are around it because if your control group is changing, if it's a minor change, and actually, it's something I have come across this a number of times in global health. It comes up particularly with cluster trials. If you're trying to if you're trying to implement a certain treatment in large clusters, like big villages or regions or what have you, that if you do it 1 year, let's say to reduce malaria or just to give antibiotics to a whole cohort of people, then the next year they won't, they all kind of get better, and it's all the concept of herd immunity and what have you. So sorry, I digressed a little bit. I think if the control group changes over time, you can have kind of two scenarios, which is that either it changes a little bit and then you don't know if it's just due to the play of chance or not, or it changes a lot, in which case you can't really use the data from before. You may say that you can do some statistical down adjustments, but I think the down weighting is going to have to be so severe that it's basically like throwing it out. Otherwise, it's going to lead to severe biases. So again, my answer would be simulation is the way to go. Simulation is not just for adaptive trials; it should be for any clinical trial planning. I think we really should get rid of sample size calculations as an exercise. It's a good preliminary exercise, but if you do a simulation, then you're much more certain that you've kind of covered all scenarios.
Great, thank you. So another related question. If you were to switch treatment halfway through the trial, how would you calculate the overall effect size in this case?
So I guess it depends on whether it's switching the active intervention or whether it's switching to placebo. So I think if it's not really something. So I think if you're switching active interventions, then it's a completely different topic. You have to talk about sequential treatment effects. That's not what this talk is about. This is smart designs where you kind of randomize designs around that. It gets pretty complex. I'm not an expert on that. I think if you were to switch the control to a treatment, again, my take on it would be more that you're not. So I guess the estimation of the control group we've already been through. So you're using historical control data. You've used that. So in the survival scenario, you'd use the first data and you'd kind of use that to augment the control data. If you're switching the treatment later on, then I think you would simply have the variable of when the patient was put on this. So sensitivity analysis would be my answer, which is like, I know probably a bit simplistic if you're looking for some complex mathematical model. If a patient's on placebo or standard of care in a clinical trial for 6 months, and then you get switched to an active intervention, let's say it's cancer, or even autoimmune disease, I'll just do sensitivity analysis. One is to treat them like they were just enrolled in the randomized trial, and one is to treat them with some sort of adjustment for they had that delay, so put that variable in. Maybe there's some other differences for that particular, maybe that particular cohort of patients look a little different than the cohort in the treatment group, simply because, I don't know, they're on placebo for half a year in the clinical trial setting. So they may have developed certain characteristics, some of the variables that are recorded. Then you can adjust for that. And I think those are the three key analyses you'd want to be doing. And if there are no differences, then no problem. There's no sensitivity. And if there's some big differences, you have to dig more into that and figure out what's really going on.
Great. Thanks for that. So now there's a kind of an analogous question, but from the data side, right? So how do you determine, first of all, there are a lot of people asking, well, how do you know you're getting a good data set? Where would you acquire one? How would you test that it's a good data set? And also, when you go to submit someone else's data set, what are some of the challenges that can arise?
Yeah, I think, I mean, the degree to which, if you're a statistician, you probably know already whether it's a good data set or not. I mean, you don't have to have experience analyzing synthetic controls data sets. You just have to have experience analyzing data sets in general. If you're not a statistician, you want to get a data set, and you have statisticians doing it. I think the best measure is probably to see how frustrated your statistician is or not. It's pretty obvious. If your team statisticians start complaining from day one, things like if you're missing a data dictionary, which actually happens surprisingly often in real world evidence. Things like that, variables that are not consistent, the amount of missingness, lack of definitions of covariates, just errors in the data. I think most real world data sets have been cleaned pretty thoroughly, but it's just the structure of it that's a bit problematic. Again, I would lean more towards, and I think this is possible in most cases, and unless you're in rare disease and you're dealing with natural history, if you can get randomized clinical trial data sets, that's really the best, right? And even the quality there differs, but again, the less your statistician complains, the better the data set probably is. It doesn't have to be comprehensive with 5,000 covariates recorded. That's also a little bit too much. If you've ever done any clinical trials, which I'm sure some of you have, you'll know that filling out 50-page case report forms is painful, and the shorter you can make those, the better, and the more likely it is that the data recording will be complete. So a lot of variables is not always a good sign, but I would get back to you kind of know it's an intuition thing. If you're a statistician, or if you're a statistician and you start complaining about the data set a lot, it's probably not a good data set. I will say, I think with natural history studies, and particularly when you're just digging around internally, like if you're a larger pharmaceutical company, you're kind of doing a scoping review, you're trying to figure out what evidence is out there. There are a lot of natural history studies that are published that maybe don't give as much detail as you'd like, and that's not the author's fault, and that's not the journal's fault, but journals have to cut it short at 2,000 or 3,000 words, so there's only so much you can put out there, and obviously folks don't want to share all the personalized information of data. So if there's no data set around that, I think you can always just contact the authors to kind of see if they're responsive and they can share some more information with you. That helps a lot in terms of figuring out the quality of the research that they've conducted as well. So I would say it's not like there's a metric that you measure this. It's more of an intuition thing. You know if you've done a couple of data sets, you know if you've done a couple of reviews. So make sure if you're doing this first time, work with someone who's done it before.
Great. So I guess the follow-up question to that was, okay, but are there, could you speak maybe to some of the maybe regulatory guidances on, what counts as a good data set, how should you submit it, and so forth?
So I'll be honest and say I haven't been part of that many submissions, and I think it's more of a, FDA and EMA releases guidance, and it's always very vague, and there's a very good reason for that, which is that I think particularly with synthetic controls, which is still new territory for a lot of people, for most people actually, every case is kind of unique. So, and it very much depends also on the priority of the disease, right? If you're just trying to get, if you're the 5th company to market trying to get indication for, some cancer or autoimmune diseases or cardiovascular disease, the priority is probably not that high, and you would have to definitely show very good data, as you would in all cases, so even if you're not using synthetic controls. But if you're dealing with something like a rare disease, particularly in the pediatric setting, you'll get a lot of leeway, because if there isn't a lot of data, there will be great, no one can argue against helping kids, right? So, there'll be a lot of leeway in terms of everyone wants to get to the same result. I think with regulatory issues, it's honestly it's more about having the conversation with the regulatory folks, instead of saying that there's no guidelines. So it's you can read the primer, and there's kind of general guidelines of what's good and what's bad data, but it's just saying the general parts of you have to have enough sample size, you have to have enough covariates, you can't have too much dropout and missingness, there has to be some consistency, you have to use the appropriate methods. It's all the standard textbook kind of stuff, but reality is that every situation is unique. The higher the medical need, the more leeway you probably, that's not to say that you can just do anything, but the higher the medical need is, the more priority that gets, the more leeway you probably get with the data. Same thing as with non-synthetic control approvals. There's not much difference there. But again, I'm not that much of an expert in regulatory affairs, so I've been part of a few, but not that many that I'd be able speak broadly about it. So just take my advice with caution on that.
Sure, and as always, there are other Cytel consultants happy to speak with anyone who has specific questions about regulatory affairs in this space. All right, so next question. Could synthetic controls be used to accelerate development and reduce costs for biosimilar manufacturers looking to launch biosimilar drugs?
I hope so. I think they could. I think there are, let's take rheumatoid arthritis as one example. There are definitely more than 100 trials out there on biologics. I think some of them might be starting to kind of share their data. You probably can't download IPD anywhere, but you can. There's some initiatives where at least you can request some analyses. So if you want to go for indications for certain subgroups. It's a little tricky because actually the control response in these trials have changed over time. There's kind of this eligibility creep, which is that patients kept getting better and better because more and more we're getting, the very bad patient, the very severe patients were the ones that were enrolled in the first trials when biologics first came around. Then there were fewer of those around because everyone started to get treated early. And then now you're kind of dealing with this moderate to mild patients that are getting enrolled in most of the trials. So I think you'd have to be a little bit careful about not taking if you were to compare something to, let's say, etanercept, don't take the trial from 99, right? Because the population is very different. But I think it's a very good case for it. And I even think there are a lot of, I know a lot of these trials, and let's just stick with rheumatoid arthritis, a lot of them have followed up for years with the patients and the treatment group. So you actually have very good long-term data on how they did. And it's kind of an extension from efficacy to efficiency, right? Where you kind of go, well, first you were randomized, but now let's continue on this treatment because you were randomized to it and you seem to be doing well. And let's just do kind of an observation that's more of a real world setting. So yeah, I think biosimilar companies should definitely look into that. So I know it's expensive to, buy 300 doses of biologics from another company, and so yeah, I don't see a reason why it shouldn't. From a regulatory point of view, I would, again, other people can speak more to this, but it's a conversation to be had. So I'd have to start that conversation, and then whatever strategy you want to apply as a company, whether it's cost or clinical benefits, you can definitely do
that. Thank you. So the next question is, when you construct a synthetic control, patients don't have a definitive entry point within an SCA. So for example, if you're conducting a study where you're looking for a population of patients failing more than 2 lines of therapy, you might get patients who enter at a time of 3rd, 4th, or 5th failure. How do you determine at what time point a patient enters an SCA?
Sorry, what? I know you're just reading from a typed question, but what time point a patient enters a synthetic control arm? I guess the question would be about the eligibility of the selection of the patients. Okay, yes. So I'm going to assume that it's about the you can type in the chat if I'm not getting you if I'm not understanding this correctly, and my apologies if I'm not. But I think the question is about determining the eligibility, and Again, it's a bit situation dependent. I know that's the easy answer. It's kind of my answer to everything. It depends. But of course, if you have a lot of data, it's very easy to match every single patient. If you have a single arm randomized clinical trial, or if you have a just sorry, not single arm, so a single arm non-randomized clinical trial. If you have a randomized clinical trial, whether you have a randomized clinical trial with fewer patients enrolled to control, there's of course eligibility criteria for that trial, and the same thing would be true if you just had kind of a case series. I think if you have a case series, you probably don't have strict eligibility criteria. But if you have a study that's a single arm study for your experimental treatment that had eligibility criteria, then of course you have to match the synthetic control patients to those eligibility criteria. And you can maybe expand that a little bit, because if you actually look at most clinical trials, there are maybe 5 to 10, oftentimes maybe 5 to 10% of patients don't actually match the eligibility criteria. They're just a little bit outside the range. So you can expand it a bit if you're really hunting for that sample size, run sensitivity analyses on that. I think the couple of things to note here, again, depends on how much data you have, how many covariates, or how much description you have of those patients. So there's not much you can do if you don't really have good data, or if it's too missing, and there's a lot you can do if you have a lot of data. So apologize again if it's an oversimplified answer, but it's not rocket science. You have to match these. Then I think what probably could, if you have a decent sample size, what kind of can bring you comfort is that again, this sensitivity analysis, you can try to expand it, you can try to narrow it, you can try to include or exclude certain types of patients that you're not sure about, and look at the sensitivities. And if none of these actually change anything, and I know it sounds too simple, but this is what we do, but if none of these change anything, then there's probably no confounding associated with what you're trying to include or exclude. That being said, of course, you can't just exclude patients because they have the same quantitative result when it's completely different patients. So there has to be some clinical common sense in there. I'm not sure if that gave a good answer. I apologize, I didn't totally understand the question, but I think at the end of the day, it's simple, but it's also tedious because you really have to match each and every patient, and you have to kind of look at all the variables that have been recorded, covariates, making sure that they match the eligibility criteria, expand it a bit, narrow it a bit, test different scenarios, see where the sensitivities lie, or where you might be able to argue that you have some robustness.
Great. And just as an aside, if we do kind of interpret your question in a different way than you had intended, please feel free to get in touch with us or with Christian after this webinar. So you'll be getting follow-up emails, and we're happy to continue this conversation afterward. So a few more questions. I know we're kind of hitting the top of the hour, but the first question is, what role did R&D quality play in the discussions and preparations for discussing study designs with SCAs and with regulators?
I don't know. That's I actually I don't. What role does research in? I think it differs from company to company. I'm not, I don't work in the R&D department. It depends on the corporate structure. I don't think I can answer that question. I can wing it, but that doesn't benefit anyone. I think it really depends on whether you're big pharma and what your internal organization looks like, and who has the decision-making power. I think sometimes it plays a big role, and other times it might not. Depends on maybe you're nimble biotech. You can make the decision. Maybe it's large pharma and certain departments are nimble, others are maybe a bit slower. I don't know. I can't answer that question. That's not for me to answer.
Sure. We'll be happy to, give that question to one of our other consultants and follow up on it. All right. So the next question is, could you talk a little bit about, the pitfalls, the major pitfalls when recruiting a control cohort, assuming that you have a high quality registry and you want a single arm trial? What other pitfalls might there be? And This person has also asked, how well accepted are synthetic control cohorts in the medical device space?
Yeah, I think about the acceptance, again, it's about having that conversation early on. That goes for synthetic controls, and it goes for any other approval. So, if you can make a good argument that there's a need for synthetic control here, if the medical need is high, the priority will be there. If you can't wait 5 years, then you have a good argument. But if you're just sort of a me too, not drug in this case, a device, then probably there won't be as much buy-in to synthetic control. Of course, the argument is saving time and money for the company that's trying to do it. It's a little different for the regulatory side. So I think really the medical need has to be there. The more that's there, the easier it is to have that conversation. Sometimes it's a matter of just really thinking it through and making the proper argument. And in terms of planning prospective registries, I think it's a bit like clinical trials. If you do them properly, if you're aware of all the pitfalls of generally designing trials, that's not necessarily, in my view, it's not specific to just synthetic controls. It's all the pitfalls in general that are around designing studies, whether it's a randomized trial or an observational study. It's like all your ethics, of course, have to be in place, and all your data recording systems have to be in place. We have randomization. You have to have a proper randomization scheme. You have to make sure that staff are probably coordinated. You get the patients you're looking for. There's proper alert systems. Data is also stored. All of those very tedious administrative issues that go into it. I really don't think there's a difference between a prospective study for synthetic control purposes versus prospective study that's trying to just answer the question of how did these particular patients on, let's say, this treatment do. If you do it well, then people will know it, and if you don't, then you're at the risk of folks asking some tough questions. So I would just I don't think there's no difference there. It's the same issues for quality.
Excellent. Okay, so final question for you. There's been a lot of talk lately about master protocols and complex innovative trial designs. Do you see these as eliminating the need for synthetic controls, or will synthetic controls become supplementary to these designs as well?
Yeah, I think with master protocols, there's it depends on what design. So if you're using a platform trial, you have an ongoing control. So those, if you're running a platform trial which drops treatments and add treatments as they become approved or fashionable or what have you, you will have an ongoing control, and then your control changes according to what's the most ethical and appropriate standard of care. So in that sense, I don't think a synthetic control is needed as much as it might have been. But if you're looking more on umbrella trials or basket trials, which often don't have a control, I think there will be an increased need for kind of running something separately, prospectively, ideally, running some prospective controlled registries to just kind of compare the intervention groups of the various subgroups or disease groups that you're looking at, and again, umbrella and basket trials. So it's to some extent it'll kind of eliminate it, and to another extent, I think there'll be an even further need for synthetic controls when clinical trials become more complex.
Thank you. So, I guess stay tuned for more answers or more research on that question, right? Because we're going to be doing several more webinars in the remainder of this year on those and related topics. All right, Christian, I just want to thank you so much for spending an hour with us and giving us this very interesting and exciting webinar. And also thanks to our audience for, I know we went a little bit over time, so it's very much appreciated. Thanks so much for joining us.
Thanks for all the great questions.
Thank you.